Hello, I'm Dr. Michael Fossel. Welcome to this webinar. We're going to talk about dementia and Alzheimer's, but from a different perspective than I think you're used to. And that is, we're going to talk about something I think a little on the optimistic but realistic side, which is the potential for finding a cure. Now, I'm well aware, more so than most, that this is the typical statement that we see with regard to Alzheimer's is at the graveyard of biotech or the graveyard of companies. And the reason for that is there have been more than 1,100 interventional trials, depending how you define them, that have failed to have any effect on the vector of the disease. So it's small wonder that people are reasonably pessimistic about the potential for finding a cure. But I'd like to give you a slightly different perspective and a novel way of looking at Alzheimer's that I think will make a difference. This is a friend of mine. Many of you are aware of Dr. Leonard Hayflick. He was the first person ever to describe cell aging. Now, a bit more than 60 years ago, actually. I still am in contact with him regularly. This is a quote that he gave me about a year or so ago when he and I were talking, and it's in the recent article that I put out. But what Len is saying is something very important, which is that we all agree that the single major risk factor for age-related neurodegenerative disease, for example, Alzheimer's, is in fact aging. But everybody then shrugs and says that's all well and good, but they entirely ignore it and move on to talk about, for example, histology and physiology, genetics, with no focus whatsoever on the major risk factor. Not only is it probably the major risk factor, but it probably offers us an innovative point of intervention that would be much more successful than anything we've looked at so far. Let me take you back about 45 years to this area. This is Stanford campus where I was trained. I did a PhD in neurobiology and then was teaching neurobiology in med school when I did my MD. In fact, they paid me to get the MD, which was convenient. But originally, I was focused on the development of the nervous system, something I found fascinating. But I became more and more entranced by what goes on at the other end of life, that is age-related disease. And what I found is that people have a very blasé attitude toward aging. They think it, quote, just happens, unquote. Sort of, you get older, what can you expect? But there's very little attention paid to the process itself. So for a number of decades, I've been intrigued by that aging process. For example, now 25 years ago this year, I gave the first talk ever at the National Institutes of Health here in Bethesda, Maryland. And what I talked about was the prospect for actually intervening successfully in age-related disease. That is, the prospect for being able to cure and prevent age-related disease by intervening in the aging process itself. That is, reversing the aging process at the genetic and cellular level. As I pointed out then, anyone who thought you could or couldn't reverse aging was being naive. The question is, where's the data? And importantly, have we truly understood the aging process, which we had not. Now, that led to my putting out the first book ever on reversing human aging, the first articles ever in the medical literature in the Journal of American Medical Association, now more than about 25 years ago, in which I pointed out that if we could understand and intervene in the fundamental cellular and genetic processes of aging at the cellular and genetic levels, we could prevent and cure many age-related diseases. That was followed by what's still currently was and still currently is the only medical textbook in this area, Cells, Aging, and Human Disease, which I published with Oxford University Press. It's an extensive look at the field, looking at each and every cell system and organ system, and the way aging affects them and where we can intervene, where we currently intervene without much success, as well as where I think we could intervene with great success. Followed finally by this book on the right for the general public, which the Wall Street Journal praised as one of the best science books of the year. And it's readable by anyone without a technical background. But the question I really asked is, have we ever intervened in aging? And the answer is we have not. We have markedly increased the mean lifespan from about a quarter of a century to a half century to currently 3/4 century over the past 200 years. But at no time have we intervened in any way with the aging process itself. What we've done essentially is deal with non-aging related diseases, allow people to live long enough to actually age. So can we actually improve lives? If I look back over the past several centuries, what I see is this. The major improvements we've made in people's lives have not been due to technical advances, for example, a fourth generation cephalosporin or the latest statin drug or robotic surgery and so on, which are interesting improvements. But the major thing we've done to improve lives has been conceptual revolutions. For example, microbial theory, which allowed us to prevent smallpox, allowed us to use hand washing, for example, during deliveries, which drastically lowered the neonatal and maternal death rates. Sterile surgeries, first rabies vaccine, Louis Pasteur, the first penicillin, the first antibiotic, that is, polio vaccine, the Salk vaccine. These are things that markedly improved the quality of human life. Interestingly enough, they not only did that and extended the lifespan you've seen over the last slide, but they also lowered the cost of medical care. That is, as an example, look at the polio vaccine. In 1950, because of the cost of iron lungs, rehabilitation care, pulmonary nursing, braces, there were actually estimates that by the year 2000, most countries would be medically bankrupt solely because of the cost of treating polio. In 2000, actually, that did not happen. In fact, WHO estimates are that the cost of preventing polio were a little less than 10 cents per patient. That didn't happen. I think we're about to undergo a second major medical revolution, and we will also find that we can lower medical care costs while improving the quality of life, and that is an understanding of the aging process. If you were to ask me, will we ever be able to reverse aging, the answer is we already have. Some 23 years ago, as I will show you in a minute, we showed the first process of age reversal in human cells in the laboratory. Some 21 years ago, that was done with human tissues in the laboratory. And again, I'll show you some of those references. The first study looking at mice was done in 2011, followed in 2012 by another study by a friend of mine that I'll show you. We're going to move on to do this with dogs and an FDA human trial aimed at Alzheimer's disease. Now, let me stress that this is not technically easy. This is very difficult. We are in the days of sort of, by the Wright brothers example, we are in the days of the Wright brothers flying machine. This is not, we're not where we need to be, but we can do this. Now, let me give you some further information on this. First, let's look at the dementias. Most of us are well aware there are many demential syndromes. These are some of the big ones, Alzheimer's, Parkinson's, frontotemporal, and vascular dementia. And while these are true clinical syndromes, there's a great deal of overlap. For example, a lot of our patients with Alzheimer's have motor deficits. A lot of Parkinson's patients have cognitive deficits. Frontotemporal patients overlap. Vascular dementia is seen on autopsy in many of these patients. So while these are accurate summaries of patient problems and accurate syndromes, there are gray zones. The borders do overlap. In fact, to such an extent in the last decade or so, we found that the alphabet soup, for example, Lewy body dementia, posterior cerebral atrophy, and so on, it not only has increased our knowledge of the diseases, but we've also found atypical dementias that don't fit in any of these categories, and mixed dementias, which fit into several categories. And that has led to a growing suspicion that what's going on is that there's something shared at a deeper level in terms of the etiology of these diseases, and they're simply expressed in different ways. And what we're looking at is age-related neurodegenerative disease, and the fundamental process is cell aging. About 2 years ago, I was invited to come to Washington for a conference I helped organize on animal models, in this case with regard to Alzheimer's disease. And the major problem in the Alzheimer's field is this: everything works in mice, but nothing works in humans. And as I think many of you are well aware of, not everything works in mice either, but the question is why? Now, part of the problem is that we share about 75% of the protein expressing genes with mice, but many of the mechanisms we're studying in translational work are based on processes that are founded in those other 25%, and it's small wonder they don't translate well. So in many respects, what we have been doing is sort of digging in the wrong place. And when that happens, whether we're digging with beta amyloid or tau tangles mitochondrial failure, or recently looking at neural growth factors, and what you find is that it doesn't matter how much money, time, and effort you put into it, you're still digging in the wrong place, and digging deeper won't help, nor will additional funds. The result still is frustrating clinical failures. And the question still is why? I was asked to write a paper as a result of the talk I gave at that conference, actually by the editor-in-chief of Alzheimer's Dementia, and asked to join our scientific advisory board. And the paper pointed out that not only do we need a model, but it offered such a model. And the model is consistent with all the clinical data we have. It's predictively valid in terms of predicting ahead of time the Eli Lilly and Biogen studies, their outcomes clinically. It also offers a novel point of intervention that we suspect is going to be much more effective clinically. Now, the result of this paper was that I got more than 600 global reprint requests and a number of invitations to speak at conferences like this one. But let me tell you a little bit about that model and what I suggested we needed. which is we needed a unified model and a systems model. The unified model needs to be able to explain the heterogeneity within dementias. That is, why do some people get Alzheimer's disease at age 25, literally, some people at age 50, some people at age 90, and some people not at all. Furthermore, why, for example, within Alzheimer's, do we see some people with more memory deficit or executive function deficit? Why in Parkinson's do we see various kinds of motor deficits? We need to be able to have a model that explains that heterogeneity. Also between dementias. Speaking of Parkinson's and Alzheimer's, why does one patient get Parkinson's and another gets Alzheimer's disease? Between species also, we know that there's a uniform cognitive decline with age in almost any species we look at. The question is, why are they expressed differently in different species? Yes, they have a different genetic background, but why do they get expressed exactly that way? Likewise, the upstream mechanisms we look at. So for example, if we look at the genetic markers, we know that there are a number of genes, which we'll come back to, and alleles that are linked to Alzheimer's or Parkinson's. The question is, how are they linked? Why is it, for example, with head injuries too, can increase your risk of Parkinson's disease or Alzheimer's? Exposure to a number of toxins like dioxin and paraquat can increase your risk of a number of these dementias. How does that happen? Why likewise infections or other exposure to radiation? Why do these translate into actual downstream clinical disease? And likewise, the downstream biomarkers. Why is it that some patients have a great deal of beta amyloid plaque, others don't? Why is it we see mitochondrial dysfunction to varying degrees in various patients? Why do these biomarkers occur at all? And why is there some degree of heterogeneity? Not only do we need a unified model, but we need a systems model. And when I say systems model, I mean that we not only need to be able to define the genes and the toxic exposures and the downstream biomarkers, but we need to understand the system itself. That is, what is the dynamic process, the cascade of pathology that takes us from upstream biomarkers or upstream mechanisms to downstream biomarkers in clinical presentations. To give you an idea what I mean, this is the cockpit of an Airbus 380. The Airbus 380 is often powered by what you see in the upper right, which is the Pearl 15 jet engine rolled by Rolls-Royce. And that's powered by that turbofan Trent 900 in the lower right, and that's a single family. Well, if I'm trying to understand how this airplane flies at 35,000 feet at hundreds of miles an hour at very high RPM and high engine temperature, I need to understand not simply a list, a category, a catalog of the components. I need to understand how they fit together and how they work dynamically. So what I need to do is understand the system, not the components. Now, that observation I just made with regard to engineering is equally true in medicine. For example, these are a list of signs and symptoms that we typically see with COVID infections, but they are not COVID. COVID is a very complex interaction between the virus you see at the right and the human immune system and the rest of the body for that matter. So if I catalog a whole series of components, that is the signs and symptoms of COVID infections, I have not understood the disease. I've not understood certainly how I can have vein disease. The same thing is true of age-related diseases. For example, these are aging biomarkers. They are at the macroscopic level, things that we see all the time: decline in the glomerular filtration rate, vascular pruning in the peripheral system, and dozens more. But these aren't aging. They are outcomes of aging, the components, the biomarkers. They are downstream signs and symptoms of aging, but they're not the dynamic process. Now, it doesn't improve it if we go down a level and look at the microscopic levels either. For example, we do see a decline in DNA repair. We see methylation changes, telomere shortening, decline in the ratio of ATP to reactive oxygen species. We see hundreds more of these biomarkers at the histologic or deep structural level, but they are not aging. They are outcomes of aging. They're things we can measure and should, but they're not the process itself. If we're looking at age-related neurodegenerative diseases, we see the same thing. So these are typical findings that we see on histology or pathology, or in some cases on imaging: glial activation, alpha-synuclein in the substantia nigra, blood-brain barrier failure, lymphatic changes, and so on. Some of these we've known, like beta amyloid, for more than 110 years, but they are not dementias. They are signs and symptoms, they're biomarkers, they're components, but they're not the disease itself. The disease itself, if we're going to understand it, means involving ourselves in why these things occur. Likewise, if we look at the genes, it's not much better. We know more than 200 genes that are related to dementias like Alzheimer's disease, but to know that is not to explain the disease. Example, you can have two ApoE4 alleles, and as we'll show in a minute, that increases your risk of Alzheimer's, but it doesn't explain to us why you get Alzheimer's, not consistently. If you have ApoE3 alleles, your risk is much lower, but we don't know why that occurs. The same is true of the rest of the genes. To identify the genes is not to identify the systemic process itself, nor to explain it. So what we need to do is understand the aging process to understand the disease and to be able to intervene effectively and prevent or cure these diseases. To do that, let me give you a quick overview on aging itself. Here are a number of species you're well aware of. Human beings typically live, for example, about 75 years. That's Coco the gorilla who spoke sign language with her little kitty, Allball. I was Coco's babysitter for a year when I was on Stanford campus. She lived about 40 years. Typical domestic animals live about 15 or 20 years, depending on the breed. Typical laboratory animals, mice, rats, and so on, usually live 2 or 3 years, again, depending on the breed. Why do they age at different rates? You can't simply shrug your shoulders and say it's just entropy, it just happens, without explaining the rationale underneath that. And it's not enough either to say they all have different genes, which is to an extent true, but it's not an explanation. What we need to do is understand how we go from genes to different rates of aging in different species. Now, as it turns out, the same thing is true if we restrict ourselves to a single species, like humans. This is a group of about 30 progeric children. About 2 decades ago, I would gather them together every year with Bill Sample there in the background, and we would take them various places around the world. In this case, we had taken them to the White House and taken them to places you're generally not allowed to go, and I took pictures of things you're generally not allowed to take pictures of. And we were having a great time until 2 children in the front row on the left, that's Sammy and Ori on the left with the baseball cap and the Hawaiian shirt, managed to set on a pressure-sensitive switch on the second step up in on the left there that goes up to the private presidential quarters, at which point the security service very politely and firmly escorted us out here to the north portico and let us take this picture. These children die overwhelmingly of age-related diseases, typically an average age of about 12.7. Almost every child in this picture died within the next few years of strokes and heart attacks, including one in the back right-hand side who died later this summer at age 7 while throwing a baseball. Why do these children die overwhelmingly of age-related disease? Why are they bald, with thin skin, with osteoporosis, osteoarthritis? We know the gene involved, the lamin A gene, but that's not an explanation. To know the gene does not tell you how you go from gene to early aging in these children. We need to dig further. Now, let's go down to the cellular level and look at some differences in cells. For example, we know that not all cells age, and we know that not all mitochondria age. Example, every cell in your body right now derives from a single fertilized oocyte. And that oocyte came from your mother, who got it from her mother, who got it from her mother. And we can, in theory, trace that line of cells back 4.5 billion years without any evidence whatsoever of aging. Likewise, if we look at mitochondria, which is involved in the aging process, as we'll talk about in a little bit, we can trace that back through the maternal line back a little more than 1.7 billion years with no evidence whatsoever of aging until you inherited it. And both the germ cell line and the mitochondrial cell line changed dramatically. There was a sudden inflection point, and most of the cells in your body now demonstrate age-related changes. So why is it that certain cells and certain mitochondria never age and others do? Well, we know that the problem is not simply one of entropy. It is one of entropy versus maintenance. And if we look at the germ cell line, what you find is that maintenance is essentially flawless in the face of entropy. The same is not true of most of the somatic cells in elderly human beings. For example, that's the hand of an 80-year-old man sitting in a wheelchair. And if I look at his keratinocytes, his macrophages, his osteocytes, his endothelial cells, if I look at any of the cells in that hand, what I find is that there are age-related changes that are consistent with a decrease in maintenance. And that is why we age. It's in some respects similar to Red Queen theory. Those of you who have sort of biological background recognize this quote from Alice in Wonderland and Lewis Carroll, in which she says, running as fast as you can is the only way to stay in one place. And that always has been true of different species. For example, the human immune system, as it battles against microbial invaders, has to run very fast to manage to stay in one place and resist infection. But the same is true within the cells, down at the deep structural level. What you find is that we are continually maintaining ourselves in the face of entropy. It's a lot of running in one place, or running as fast as you can, just to stay in one place. As an example, let's look at molecular pools. If we look at any molecular pool, for example, the collagen and elastin in my face, the beta amyloid molecules in my brain, myoglobin, any of the molecules in my body, what you find is they're continually being turned over at varying rates. But as we get older, what happens is the rate of turnover is lower and lower. So in young cells, they run very fast to stay in one place. They're continually recycling molecules, and we'll look at an example of that in a minute. So anytime you have damage, signified here with a little red star, what you find is that it's washed out. You're continually replacing damaged molecules with new ones, and the outcome is generally in young cells, cells are very functional because the molecule is very functional. But as telomeres shorten and as gene expression changes as a result, what you find is molecular turnover slows, and older cells simply are less likely to recycle molecules as quickly. So you begin to get a gradual accrual of damage. And this is even worse in the oldest cells, when the result of that is not only molecular dysfunction, but cell dysfunction generally, cell necrosis, cell failure, tissue failure, and age-related disease. You can look at it mathematically. This is taken from my textbook. And essentially all this says is that if you have a rapid rate of turnover per unit time, then the accrued damage percentage at equilibrium is very small. And if you have a low rate of turnover per unit time, then over time, the accrued damage at equilibrium will be very high. Now, let's look at an example of this within an organ that is appropriate for this webinar, and that is the human brain. In this case, we're going to restrict ourselves temporarily to beta amyloid turnover, which is by no means the only case involved. We can measure beta amyloid turnover in the brain. What you find is that in young cells, microglia, for example, they are continually producing beta amyloid, then secreting it, rebinding it, internalizing it, degrading it, producing it, secreting it, and it's a continual recycling system. But as we get older, what happens is the rate of turnover slows a bit. And so anytime there's accrued damage, the percentage of damage goes up. The percentage in this case of beta amyloid microaggregates go up. And you might ask reasonably enough what this has to do with different genetic risks. For example, if I have two ApoE2 alleles, what you find is my molecules, my beta amyloid molecules, are very unsticky. They don't tend to cross-link and degrade very easily. So I have very few in the way of beta amyloid microaggregates as I get older. Not none, just a lot fewer than I have, for example, if I have two ApoE4 alleles, because the molecules produced here are stickier. They tend to produce a higher rate of damage production. And the result is you get more in the way of beta amyloid microaggregates if I have two ApoE4 alleles, which is why these patients are more likely to have Alzheimer's disease and at an earlier age than patients with, for example, ApoE3 or ApoE2 alleles. In any case, regardless, what you find is that as we get older, the rate of turnover of beta amyloid molecules in the microglia goes even lower and lower, and we've measured this. And the result is beta amyloid plaque. Now, this is not simply true of beta amyloid. It's equally true if we're looking at tau tangles, mitochondrial dysfunction, mitochondrial enzymes, inflammatory changes, microglial activation. Almost anything we look at, what you find is it goes up as we see cell senescence begin. The result of that, obviously, is a pruning of synaptic trees, cellular dysfunction, necrotic neurons, and what we see clinically as one of the dementias, expressed in different ways because people had different genetic and epigenetic backgrounds. This is sort of an overview of what's going on. You have upstream a number of risk factors, downstream a number of outcomes, biomarkers, if you will, and then a cell senescence process in between. Now, the upstream risk factors include things we can't do much about, like chronologic age, but also genetic risk, which we just alluded to, for example, ApoE4 alleles versus ApoE2 alleles, exposure to chemotherapy, which increases your rate of cell aging, certain toxins, for example, again, dioxin and paraquat are classics, other toxins, whether heavy metals or otherwise. Head trauma is a classic example that increases your risk for a number of age-related dementias. Hypertension and hyperglycemia increase your risk for vascular dementia. And we can go on with many other examples of this. Downstream, what we see are the kind of things that we alluded to earlier, kind of measurements we can make that we find in dementias and many other age-related diseases. But between these, what we see is that the upstream risk factors increase the rate of cell division, which causes telomere shortening, which causes gene expression changes, which causes slowing of the molecular turnover, which causes cell dysfunction, failure, and cell death. And the outcome is what we see downstream. Now, the question is, can we intervene? Well, upstream, there are a number of things we've tried to intervene in. For example, I do know that if I can normalize your blood pressure, make sure you're euglycemic, I lower your risk for vascular dementia. But I don't lower it to zero. I just postpone it. The same is true of head trauma. If you have no head trauma, you have a lower risk of many of the dementias, and often at a more advanced age, but not no risk. So all of these things are partial ways of intervening. As I say, medical intervention with regard to hypertension and diabetes, for example. Downstream, likewise, we've tried to intervene. For example, beta amyloid, the use of monoclonal antibodies to interfere with beta amyloid plaques. But none of these have been shown ever to markedly influence the vector of the disease. But I think we can do this if we intervene directly at the central process, which is cell aging itself, and shorten telomeres with altered gene expression. The reason I think that is that it works. The first case that we were ever able to show in human history that we could reverse aging was done in human cells in the laboratory now some 23 years ago by Andrea Bodnar in this classic paper with her colleagues at Geron Corporation in the Bay Area in San Francisco area. And what they did was they took old human cells and showed that when you use telomerase, you can reset the pattern of gene expression to that of young human cells. Well, turns out you can do the same thing with human tissues. This was done by Walter Funk and his group at the same institution in Geron Corporation in California. And what they did was show that you can actually grow young human skin from old human tissue, old human cells, if you use telomerase to reset them. So you can literally regrow young skin from old skin. The same thing turns out is true of bone cells, that is osteocytes. The same is true of vascular endothelial cells. So you can literally grow young vascular tissue from old damaged cells in the human vascular system in the laboratory. Can we do this in animals? The first time that was ever done was in Ron DePino's lab in about 2011. The second case is this one, and this was done by Maria Blasco's group at CNIO in Madrid. And she and her colleagues used a technique that we could actually use in human cases, which is using gene therapy to put in telomerase. What you find is that the telomerase improves the physiology and the behavior, as measured here, for example, by bone mineral density, subcutaneous fat, and so on. It also increased the lifespan. Now, this technique is difficult. It is daunting, in fact, and we're not very good at it, but it works. So the question is, what can we do to intervene with human age-related dementias? And the answer is, most companies, for example, Lilly and Biogen, have focused on amyloid. Some companies have focused on tau tangles, some on both. Some on mitochondrial dysfunction, some on immune problems or inflammatory problems. Some also on things like nerve growth factors. But none of those have worked because there are many, many things that happen downstream from cell aging. What we're going to do is target cell aging itself, the underlying cause of all these different pathologies and dozens more. What we're going to do is take this to human trials to show that we can restore young cell function in aging cells, actually in vivo. We will be intervening using telomerase therapy. Our first target will be Alzheimer's disease, but this should apply to other age-related neurodegenerative diseases and in the long run to other age-related diseases generally. Our major focus will be not only the safety and making sure that we can do this carefully and right, but efficacy, which is difficult. And finally, an issue which you might not think much of, but that's credibility. We are well aware, as I've said, that there have been more than 1,100 interventional trials which have uniformly, by global consensus, failed to have a significant impact on disease course. So if we suggest that we can not only slow, not only stop, but actually reverse some of the cognitive decline in age-related human dementias, we want to be very careful that the data speaks for itself. Hence, we're going to be very careful in what we do. In short, I think that this issue, dementia and Alzheimer's, whether we can find a cure or not, the answer is yes, we probably can. And I think we can demonstrate this within the next 5 years to the satisfaction of all involved. I certainly hope so. I hope you'll join me in wishing us the best of fortune in trying this. And for those of you who have any questions about what we can do, I'd be more than happy to talk to you further. You can contact me at this email address. In the meantime, I hope you enjoy the rest of the webinar, and I wish you best of luck.